Very good morning to everyone. This is Sandeep here, welcoming you all in this ongoing annual Distinguished Lecture Series 2021, organized by Center for Health Science and Technology, JIS Institute of Advanced Studies and Research, Kolkata, in short, JIS IASR. So today we will be having our fourth lecture out of the 6 lectures scheduled throughout this month of August in order to celebrate the 2nd anniversary of our center as well as the institute. Before we dive into the science, a few information regarding our center. The CHEST offers a master's program in medical biotechnology and bioinformatics, along with 1-year advanced diploma in bioinformatics, as well as the PhD program. The PhD program is primarily based in the field of medical microbiology and bioinformatics. For more details, please visit our website, jisiasr.org, or you can email us or call us at the contacts that you can see in the screen. Our admissions are going on right now. So this lecture series is based on the contributions of medical biotechnology and bioinformatics towards a better world. And today, we are really honored to have with us Professor Evgeni Sokurenko, who is a luminary in both the fields. I am fortunate to have worked with him for 5 long years. I still cherish those days. Thank you so much for accepting our invitation. Without any more delay, I request my colleague Sujoy to take the stage and introduce our alma mater. So one request to all the participants, if you have any questions or comments during the lecture period, as well as at the end, you are welcome to write them in the chat section of the Zoom and comment section of the YouTube, where we are going live right now via our YouTube page, CHEST JIS IASR. All the comments or questions will be addressed at the end of the talk. So over to you, Sujoy.
Thank you, Sandeep. Good morning, everyone. This is Sujoy Chattopadhyay, and I have the responsibility of introducing today's distinguished speaker to you all. Well, I can speak about him for the next 24 hours non-stop. So kind of confused where to start and where to end. To begin with a few pleasure words, it's a great pleasure. I'm delighted. I feel honored to have him with us. Dr. Evgeni Sokurenko. He is a professor of microbiology at the University of Washington, Seattle in the US. In 1985, Dr. Sokurenko received his MD degree from Moscow Medical Sechenov Academy. I believe the specialty was infectious diseases and epidemiology. Then in 1989, he received his PhD in medical microbiology from the Institute of Genetics and Selection of Microorganisms in Moscow. Afterwards, he did his postdoc in the School of Medicine, University of Tennessee in the US in Memphis, Tennessee. Finally, in 1999, he joined as a faculty at the Department of Microbiology in the School of Medicine of University of Washington in Seattle, where I was fortunate enough to join his laboratory in 2003 as a postdoc. Dr. Sokurenko's primary research interests are the molecular evolution and population genomics of microbial pathogenicity and antibiotic resistance, as well as the molecular mechanisms of bacterial adhesins that help bacteria in the attachment to host cells and tissues. The main focus is on the extraintestinal pathogenic E. coli and escape pathogens. For those here who don't know what escape stands for, it's an acronym encompassing some gram-positive and gram-negative bacterial species imposing tremendous threats on human civilization as defined by World Health Organization. E stands for Enterococcus faecium, S for Staph aureus, K for Klebsiella pneumoniae, A for Acinetobacter baumannii, P for Pseudomonas aeruginosa, and another E for Enterobacter species. Dr. Sokurenko has served on over 50 review panels of the NIH, that's National Institutes of Health in the US, and published over 150 papers and book chapters. He is a founder member of IG Genomics, a molecular diagnostic company for infectious diseases, and of a nonprofit 5013 organization, Armada, Antibiotic Resistance Monitoring, Analysis, and Diagnostics Alliance. For me, he has been my Seattle dad who taught me research, science, and philosophy, who taught me independence, who taught me how to take vodka shots. I mean, so many. And it doesn't mean that I didn't know anything of those things earlier, but he refined my ideas, my thoughts. I'm being emotional. However, in return, I tried to teach him one thing, just one thing, that is to pronounce my last name. And he appeared to be an extremely bad student in this regard. Okay, enough of trash. Let's hear him. Evgeni, the stage is yours.
Well, thank you very much. Thank you very much, Sujoy. Thank you very much, Shandip. Yeah, it's so nice to see. So nice to see both of you. As Shandip said, he was 5 long years, and Sujoy, what's the last name? I don't remember the last name. Yes, Sujoy. Yeah, still have problems. Was for much longer, and these were fantastic years we spent together, truly as colleagues. We learned from each other, and I certainly cherish this wonderful time we had. So as Sujoy mentioned, my primary focus are clinically significant bacteria, but I regard myself really specializing in 2 wide areas. One is molecular evolution and molecular epidemiology. So evolution, epidemiological, evolutionary epidemiology, I would say, of microorganisms. And another one is protein structure and function. And my primary focus was always on adhesins, on bacterial attachment proteins. So this actually was quite naturally for me to shift slightly my attention, or to expand my attention to the pandemic virus, SARS-CoV-2, and specifically to its molecular epidemiology, evolution, and the way it's attaching to human cells. And as Sujoy, of course, Shandip remember, I always was fan of the smallest possible, finding the effect of smallest possible changes, mutations in one amino acid that could change the function and affect the virulence of microorganisms. So that's how I started to work on mutations, emergence of mutations in the SARS-CoV-2. Okay, so I would like to start. For this, I would need to share my screen, but someone has to allow me screen sharing. So I need to be a co-host to do so. Okay, now let's try it again. All right, fantastic. Now it seemed to work. Okay. And let's put it this way. Okay, so does everyone see my full screen?
Yes.
Okay. Okay, wonderful. Let's start. Title of my talk would be Evolution of the SARS-CoV-2 and about the global impact of small changes, of the mutations there. I just will go quickly through some basics. SARS-CoV-2 comes from severe acute respiratory syndrome. Coronavirus is its original name was actually 2019 novel coronavirus. Yeah, but then it was changed to SARS-CoV-2, and it's number 2 because number 1 of SARS-CoV, just SARS-CoV, was the one that caused the SARS epidemic in 2002 to 2004. So next would be just to remind you guys that viruses are not alive. And actually, there is bacteria, eukarya, us humans belong to eukarya, and archaea, another type of microorganisms. And viruses really do not consider to be alive because they don't have any type of their own metabolism. But most important for us to know that viruses are everywhere. They affecting every living organism, whether it is bacteria, prokaryote, is it prokaryote or eukaryote? Yeah, everyone has their own viruses. And viruses evolved many times in the process of evolution independently from each other. So there is no single origin, evolutionary origin of viruses. Just to tell you how flexible is this system to adapt to very different living organisms multiple times. So this is a tree of known viruses, and again there is really no common origin for them. And the most critical thing to show you that when the COVID-19 pandemic started, there were lots of talks. So it looks like influenza. It behaves like virus behaves like influenza virus. We really know now it's not true, and that really the influenza viruses has nothing to do evolutionary with coronaviruses to which SARS-CoV-2 belongs. They're completely independently emerged, and whatever similarity between them, it's really not due to their common origin. All viruses can be divided on 2 big families. DNA viruses, the ones that contain DNA as their genetic material, and RNA viruses that use RNA as their carriers of their genetic material. So RNA viruses is a huge group, and again, influenza belongs there, and some other viruses, as you can see. And coronavirus, including SARS-CoV-2, are RNA viruses. What is relatively special about the coronaviruses are that they're relatively big to compare with other RNA viruses. Their genome is 30 kilobases. And interestingly, unlike other RNA viruses, their replication is relatively accurate. They do not accumulate too often mistakes during replication, and because they have some kind of proofreading system. Anyhow, that's overall about the coronaviruses. Let's go more closer to the SARS-CoV-2. A little bit more. Coronaviruses, family of the viruses, there is 4 big families: alpha, beta, gamma, delta. They infect different animals, and sometimes different types of coronaviruses will infect the same animal. And in humans, we have multiple coronaviruses besides SARS-CoV-2. I already mentioned SARS-CoV-1. There was MERS virus, as you remember, but there is also common cold coronaviruses that we have, and that causing about I don't know what estimates are, 30 to 50% of common cold cases. All right, so when the SARS-CoV-2 emerged, of course, the big question was, okay, where it came from. And when the genome was obtained from Wuhan, the first strains came from China, from Wuhan. So it became very clear that the closest coronavirus known then was from a bat, isolated from a bat. And this is phylogenetic tree where you see its close relatedness to a bat coronavirus. By the way, a couple other coronaviruses. First of all, the SARS-CoV-1 itself, you see it's also originated seemingly in bats, but they are not related to the SARS-CoV-2. It was independently emerged virus. Common cold viruses actually seem to emerge and jump to humans from other animals like mouse or cow. That's where the origin. And the MERS coronavirus, another pandemic, one also more related, actually not related to closely to any of other known coronaviruses. Okay, SARS-CoV-2 emerged really independently from other viruses, as we know. A little bit of structure, about structure. There is a spike protein. That's actually what makes coronaviruses to have the structures, coronal-looking structures, and we will talk a lot about it. It's so-called enveloped virus. It has a membrane from a human cell. There is some other proteins, important. Some of them are on the surface, but by far the biggest protein on the surface and the biggest antigen is the spike protein. And here is, if you look at the genome, spike protein truly is the second largest gene after the main open reading frame that codes for 16 different proteins. This is where the spike protein gene is located. All right. A little bit more about similarity with SARS-CoV-2 with bats. And this is the level of similarity. So 100%. So the closest, again, similarity-wise to the SARS-CoV-2 is one of the bat coronaviruses that we isolated. What is important for you to notice here, the spike protein gene is actually one of the most diversified genes in coronaviruses. You see, it's the least level, about 90% identity with the closest known virus. And there is lots of diversity to compare with other coronaviruses for the spike protein. And we'll talk a lot about this diversity, why this protein specifically is so diverse. So what does the spike protein, what is the function? Of course, we know well now, it binds to human cells, and specifically it binds to so-called ACE2 receptor. And what is ACE2? ACE2 stands for angiotensin converting enzyme 2, which main function is actually to balance effect of another angiotensin converting enzyme so that our blood vessels are not staying too constricted, too narrow, or too wide. It's a very important enzyme on the surface of the human cells, and it's located across many, many different tissues. Upper respiratory tract has lots of them. Lung, alveolar cells, Small intestine enteroviruses has a very high level of ACE2, and it's actually, coronaviruses, especially in its natural environment, seem to be enteroviruses. That's where actually they're primarily where they are circulating in the intestine. Yeah, but it's a different story. Yeah, we studied a lot whether or not it's significant for human coronaviruses. Does not seem to be, actually. Yeah, okay. So now let's talk specifically again. What another interesting thing about evolution of the coronaviruses. There is a hypothesis, it's not really fully proven yet, that the SARS-CoV-2 actually has small regions of recombination, small pieces of DNA inserted from pangolins. So that it is a result actually of two different coronaviruses to recombine, and that's where the region really of the ones that seem to be coming from the pangolins. There are lots of talk, of course, as you know, whether or not it is of a lab origin or not, and there is evidence for it, evidence against it. I will not talk today about this anymore. Frankly, I see it's possible that it is of lab origin. I don't think how much it could help us right now, and I truly believe that certainly, even if it's of lab origin, it certainly was not created as a pandemic virus. We just don't have right models to create pandemic viruses, and if it escaped, it was truly an accident. But anyhow, that's all what I would like to talk about that issue. Now, it's just strikingly again that there is a quite diverse small region in the spike protein gene that even people are starting to think that maybe it came from a different animal. This region truly encompasses, and I will be showing a lot this slide, one of the most variable and functionally important part of the receptor binding domain of spike proteins, the one that actually binds to the ACE2 receptor. So overall, there is the spike protein has two big regions that are sticking out. Its N-terminal domain we will be talking about, receptor binding domains that binds ACE2, and so-called stem domains that attach to virus, and then after the receptor binding domain binds to the cell, these two domains are being cleaved out, and the stem domains perform the fusion function. So remember this picture, I just will be showing you again and again. This spike protein actually is a trimer, as you can see here, and this is a top view. So it's a three proteins that are intertwined together, and that's what actually gives it this mushroom or corona-like shape. I will tell you some mysteries about coronaviruses that still exist. One is that the receptor binding domain of the spike protein, the portion that binds ACE2 receptor, it has to pop up from the spike trimer. So whenever it's in the closed conformation, which is a regular one, it cannot bind the receptor. It should pop up before it can bind. We still don't understand. We understand very well. We know so many structures. We still don't understand what exactly triggers the pop-up of the receptor binding domain. Yeah, we know mutations that, I will tell you, that promote this pop up, but we don't still understand what is the natural trigger for it to going from down to up conformation, and that's unfortunately restricts our ability to design good antibodies, for example. Another mystery about the coronavirus SARS-CoV-2 is that actually it's by far not the only virus that binds ACE2 receptor on human cells. Others, for example, the other SARS, the original SARS virus or MERS virus, or even the common cold viruses, coronaviruses, bind ACE2 too. So by itself, it doesn't seem to be the only, or maybe even the main explanation why this particular virus became pandemic. Yeah, there is evidence that the affinity of binding is higher. Whether this is truly the key explanation, we still cannot say for sure. Number three, mystery about the coronavirus, SARS-CoV-2. As I told you, the spike protein, after it binds ACE2 receptor, it has to be cleaved. Only then the fusion of the virus happens with the human cell. So as there is well-known protease, TMPRSS2, that is common among other coronaviruses that does perform this cleavage function. It's actually a human protease. In SARS-CoV-2, there is another protease that comes also from human, furin-like protease that also cleaves. So there is actually two proteases that cleave the spike protein in coronaviruses. And you know, the site of the second protease, people speculating that yes, someone inserted it in the lab, that it's because it's so unique among coronaviruses that someone inserted it from a different virus into the SARS-CoV-2. It's true that other viruses, some other viruses, but not coronaviruses, do have this furin-like protease. We still don't understand really why it is so critical for whether it's very critical for virus to have the two proteases. And finally, the mystery, I told you there is so-called N-terminal domain, the one that precedes the receptor binding domain in the protein. It's sort of sticking out on a side of the protein. We don't know its function. In other viruses, we know that actually it's a lectin, it binds sugars, and specifically sialic acids, among other coronaviruses. We don't know yet the receptor for the N-terminal domain. And in my opinion, this is actually very critical, could be very critical, because this could be the trigger how the virus originally attaches to mucosal surfaces, and maybe the trigger why the receptor binding domain is popping up. This is my hypothesis based on studies of other bacterial attachment proteins. Yeah, so all these mysteries are still existing, and it's very interesting to find out what will happen. Now, let's go to mutations. After it emerged, very soon became clear that yeah, it's mutating. It's not mutating very fast, but mutations do accumulate, and mutations do accumulate in the spike protein gene. It's changing its structure, and this is an old, really old next strain phylogenetic tree from the nextstrain.org. I highly recommend you to visit if you didn't yet, this site. It's a very, very beautifully made public site about phylogeny of many different viruses. So, and that's every color corresponds to particular type of mutation, particular type of clade. And here are different few mutations that emerged during the evolution. Again, it's quite old slide. Everything becomes old the moment you talk about coronavirus, because every day, literally, 100 new papers will be appearing on it. But the very first mutation was a change of aspartic acid to, in position 615 of the coronaviruses, to glycine. This was the very first mutation that seemed to emerge even oh my God, I lost the emerged even in 2019. This mutation, specifically, what was found, what was the effect? It actually made the spike protein to go in the up conformation much easier. It is far away from the receptor binding domain, but it is actually popping up allosterically, making the receptor binding domain to pop up more easily. That's considered to be was a selection for this mutation very early. If it's going up easier, so then it binds to ACE2 receptor easier. All right. The second mutation, it was mentioned in this review, was a change of asparagine in position 501, and most of changes was to tyrosine or threonine. And now this mutation actually was located in the interface between the receptor binding domain of the spike and the ACE2 receptor. So lots of structures, again, lots of them obtaining together with the receptor binding receptor itself. So we understand a lot what is the interface between the two proteins. This position is not mentioned here. It was from a different paper, but just showing very nicely how well we understand how the spike binds to the ACE2. Specifically, the 501 mutation affecting a residue, asparagine again, right in the interface. Here is the asparagine. Yeah, and it's thought that actually this mutation makes the binding stronger of the to the ACE2 receptor, and it also affects ability of antibodies that able to neutralize spike protein, because it will bind to this binding pocket. It makes it less sensitive to this type of antibodies. And originally, this mutation, you see, it emerged in so-called, you remember, British variants, South African, Brazil variant. You can see it in many different variants. And it was shown that it can help to escape the antibodies. So big mutation. That's actually was pretty much mutation that we're starting to pay attention to, like mutants can emerge and might mean difference. Another mutation from this great review was a change of leucine to arginine. So this L452R mutation was one of the latest mutations that emerged. And that was a mutation that actually we almost by accident started to pay attention, specifically my lab. The leucine 452 is actually located close to the interface where the spike protein binds to the ACE2 receptor, but it's not right there. It really sticks out on the side of the protein. And yeah, I'll talk a little bit more what could it mean. But it's unlike, again, 501 that is right in the interface, this is sticking outside. I showed you this slide before about this highly diverse, important regions within the receptor binding domains. There is so-called loop epitope region and receptor binding reach itself, the interface. And that's what's thought possibly to come actually from a different a virus from a different animal, from pangolin, not from the bat. But again, this is still controversial. Yeah, the interesting thing was for us that actually vast majority, if not all mutations of interest, current interest at least, are located in this region. So what we did, we amplified this region 500 base pair, and Sujo and Shandip remember well, that's a very good size to amplify, because you can then sequence it just in one run, and it's used a lot in bacterial typing. For example, these different regions of 500 base pairs or less. So we amplified that region in 550 viruses in positive samples. We got clinical samples. We published it again. Yeah, we published it just recently. Was accepted in Journal of Clinical Microbiology. Let me turn off this phone. Okay. Yeah. So let's say what we got. We, in this 550 isolates, we got lots of isolates with mutations. Many mutations were so-called silent mutations, didn't change the amino acids, but we found quite a few mutations that did change their amino acids. And if you will look carefully, vast majority of these mutations are in some kind of functional, located in functional regions that are of important for function. These mutations were found again and again in different viruses, as we know now. So these are truly positions that are hotspot mutations for the viruses. So when we look across our collection, yeah, I really apologize now. Sorry. Yeah, so we found in this collection that the L452, the leucine change to arginine, is the most common mutation that we see. Certainly was the most dominant to compare to other mutations. What we knew then, this mutation just got attention, and it was we're studying in February, got attention because it was found among viruses isolated during California outbreaks back in January, February. Was known then that this mutation does occur in this particular region. What was also known, and it was known for, they called it Cal20C variant then, belonging from a more defined better defined clade B1429. It's actually 42729. Now it's called, when they started to use Greek letters, an Epsilon, variant Epsilon. So we knew that this mutation is present in the ribosome binding site of the protein. But we also knew by then that there are two other mutations in the spike protein of the Californian variant, both of them located in the N-terminal domain. So the N-terminal domain itself also has a hotspot for mutations. For example, this change of tryptophan in 152 position to cysteine has been known before to be a hotspot for mutations. So N-terminal domain also functionally important, and antibodies against this particular epitope in it are also very, very important for protection against the virus. So we went back to our collection and we amplified the N-terminal region, another region in the spike protein that comes from N-terminus, and look what kind of mutations. And indeed, we found these two mutations typical for the California strain. We found in majority of the strains that was carrying the L452R mutation. In nearly all, but we did not find it in two strains. And this was quite puzzling for us. Okay, it just emerged, we know about the outbreaks, but there is some of them actually do not have the commonly occurring co-mutations. We took some strains from our collection and we sequenced the entire genome. And we took strains that carrying the L34R mutation together with the N-terminal mutations and those that do not carry it. And indeed, they split to completely different groups of strains. They were absolutely not related to one another. So it became clear to us that we have some strains in hands that are different from what was causing the outbreaks in California. The Epsilon variant is called right now. So we pulled out lots more genomes from JSA database that has where all or most of the genome sequence genomes going to, and we saw that really our strains without the mutations are completely different variants that are mutation that cause the entire group to expand. So it was really important mutations that allowed the entire group to emerge. When we look closely to the Epsilon Californian strain, we also found that the mutations in the N-terminal were appearing before the L452R, but they never resulted in a big expansion of the virus. It was again the L452R that was the trigger for the emergence and becoming a really outbreak strain. So this mutation indeed was evolutionary trigger for expansion of these variants. By the way, so our variants, which we call Cal20A, then it was the only case where what was recorded that gorillas in San Diego Zoo got COVID-19. And that was the virus that caused it. So we looked further. So now, again, back to the L452 position. It was shown that it seemed to improve the binding affinity to the ACE2 receptor. It certainly allows escape from some neutralizing antibodies, but really still now, there is so many papers now about this mutation. We still don't know exactly why this mutation has a selective advantage. This mutation happened to be selected in so many different variants across the globe. And really starting from December, the earliest of the 2020, and especially in February, in March, you can see how many different clades, different variants started to accumulate the mutation in this position. Vast majority have the mutation to arginine, L34R, but some of them actually have to another charged amino acid, to glutamic, to glutamine, the Q. And so interestingly, the mutation in Q happens in so-called lambda variant. Okay, so here is the Californian Epsilon strain. Our group, which we found, actually never expanded big. So we actually could not find many natural representatives. And here we just point to where it would be located if we will find. But we used it well, this variant. We found that it is under selection. Interesting thing. So it's clear evidence that this mutation is under positive selection, emerging again and again. And almost all recent variants of concern of interest has the mutation. And of course, very interesting that these guys have the mutation, the Kappa variant and the Delta variant. And by the way, in the parentheses, it doesn't show the origin, it shows where the most of the isolates were coming from at the time we analyzed. So we know about Kappa, and we know about Delta, and we believe it came from India. At least some evidence points to that. But both of the variants carry the L452 mutation. So we look more closely into this. When did it emerge? And this is a big tree that we recently, a few weeks ago, pulled out genomes from. And we looked, so this both Kappa and Delta, they're cousin variants, they're very closely related, and both of them forming this B1617 clade of the virus. Okay, let's see what kind of mutations were acquired when in this group. The very first mutation actually for the big clade was a proline change to arginine. Proline change to arginine in position 681, which is far away from the receptor binding domain structure. So it's actually close to where the 614 mutation was happening. And mutation in this particular position was noted before, specifically so-called UK or British variant. We found it really first time we started to pay attention to this. We still don't know what exactly it does. We know it's affecting ability of furin-like protease to cleave the protein. It seems to enhance it. We don't know exactly how it's affecting the virulence of the virus. But this was pretty much seemingly to be the first mutation that came in. By the way, it never resulted, it came in, but it never resulted in expansion. You see this little n, it's a number of genomes that were in the database, just one genome we found. with this mutation. What's the next mutation? It's our L452R mutation. That was the second mutation that was acquired in the entire clade. So it was truly one of the founding mutations for the entire clade. You also notice that after it emerged, it also didn't result in a big expansion of the viruses. What did help to the expansion was mutation of the glutamic acid in 484 position to glutamine. Simply very similar amino acids. But that was a mutation that was first connected truly to the expansion of a big clade of the viruses. Now it's called a Kappa variant that emerged originally as a big variant circulating in India and causing the pandemic there. This position, the 45484, also located right where spike protein binds ACE2 protein, ACE2 receptor. It's right in the interface as well. And we knew about this mutation, at least in this position, to a different mutation. It was already circulating in some other variants that originally emerged. However, this mutation was a different mutation, and also this mutation did not happen in the background of the L452R mutation. So now, what happened next? Where is the delta? Here is the delta. It's a mutation of the threonine in 478 to leucine. That's really what triggered expansion of the Delta variant. Very interesting. Though this mutation came after the 484 mutation, it seemed to replace it. So it didn't co-emerge. It's possibly that there was a recombination that really swapped the region and resulted in it. Some weird events happening here, because in the middle of this clade that was called Delta clade, and now is truly expanding everywhere, there is this region that seem to be mixed up mutations together. Very few strains, just one genome, maybe mistakes, genomic mistakes, maybe not. Maybe these particular strains give us clues how did it emerge. So I wish that Sujo and Shandip are still here to help me to figure out what really happened, how the Delta emerged from Kappa. Or maybe they can figure it out by themselves right now. But nevertheless, this is our latest results, very interesting events, how the Delta emerged from Kappa. Okay, the 478 mutation was known before. Again, we had seen it even in our collection back in February, and you can see some Californian strains different from the L42R have it, but just by itself, again, not in combination with the leucine 452 mutation. So where is it? These mutations that may be critical for the emergence of Delta, it's actually also located close to the interface. But like L452R, it is not exactly in the interface. It's actually in the loop, inflexible loop near it. So it's not really affecting directly the binding of the ACE2. It's known again that so many papers coming out, and it's hard to understand what really is the functional effect of the mutation. Does it changing the affinity of binding to the receptor or not? it does seem to allow escape of some neutralizing antibodies. We don't know. What we're trying again to figure out is that a combination of mutation may be not really random, but really core selected. At the end, what really is my opinion, personal and supported sometimes by opinion of others, what will happen? It looks like unlikely virus will go away. It's clearly now that it can adapt and keep circulating, can actually reinfect people. We hope that it will be circulating and reduce its virulence and maybe become a cold-like, common cold-like virus with humans being reservoir there. It may retain the virulence and maybe it will jump to a different host, who knows, dogs, cats, whatever, who are circulating around the humans. A nightmare scenario, unfortunately, seems to be unraveling, that vaccine escape variants starting to appear, and Delta, the success of Delta may be in part due to the fact that it can infect vaccinated people as well as non-vaccinated. It's not causing a severe effect there, but it can. The problem is that if it starts to evolve among the vaccinated people, we don't know what direction it will go. If it will go towards increasing the virulence in vaccinated people, we are in real trouble. The real trouble is that it actually will not be that easy to correct the vaccine itself to induce antibodies to that particular type of variants, because we already will have antibodies against the rest of the spike protein, and so-called effect of the original antigenic scene, so-called, will prevent reusage, easily modification of the existing vaccines. We'll have to come up with a new generation of epitope-specific vaccines. Okay, so that's my acknowledgement slide. I certainly, most of the work done here was done by Veronica Chesnakova, and both Shandip and Sujoy should remember her well. Yeah, Dagmara Kishela, Lydia Larson contributed a lot to the analysis. ID Genomics, the company that was founded, a startup company founded about 7 years ago, contributed significantly to the study. Institute for Environmental Health really provided lots of isolates and provides genomic sequencing service for us. Yeah, and Kaiser Permanente also provided us lots of clinical strains for analysis. So here will be the end for my talk. Thank you very much for listening, and I'm ready, if I still have a little bit of time, to answer your questions.
Thank you so much, Evgeni. It's always equally delightful to hear you, because it's not like the normal talk. I mean, it's you deliver the talk like a teacher. I mean, who actually explains everything to the students. So I just consider.
And I give it with a strong Russian accent. So that makes it different.
Yeah, and it's kind of coming to me. I mean, from Russian to English and to Bengali. I mean, it takes a lot of time to process in my brain. Yeah. So, well, others are probably adding some questions here. I have a couple of questions. One is that you showed in the tree, I mean, deep branches showing the origin of first, I mean, in the background of mutation at position 452. First one was 484, and then which yielded a kind of a big clade called Kappa, and then there is the mutation 478, where actually you said the 484 mutation got lost. So would there be any possibility that in the background of 452, this 478 and 484, emerged along two different directions? Why? What is the basis of thinking that Kappa should stay there as an ancestor? That's
that's a very good. Yeah, that's a very good question. If you look at the tree, it does seem to. I can share it again. Yeah, if you will look at the tree, it's clearly more closely related. There is a big clade of Kappa, but there is a smaller clade, and it's called clade 3, so-called, of Kappa that is more closely related here. And, there is some common mutations between them. And
but
as
could it be the result of recombination that we see there? In those variants.
This certainly could be a recombination. Again, we did not have yet time. This is analysis that we did not publish yet, and it really happened last week. We started to look, and there is indeed some very strange isolates that we see within the tree. As you know, well, it could be mistake of the sequencing. It could be some aberrations of the tree building algorithm. So, but that's why, it would be so good, Sujoy and Shandip, for you to take a look at it.
Yeah, the other thing is when you talked about this 452 mutation, you told that in the Californian variants, you found a couple of other mutations which appeared to be the ancestral ones happened in the N-terminal domain. And
Yes.
then you showed tree where we found this 452 mutation. It's this 452 is a hotspot. There's a convergence happening from all different directions. So, but you don't see the same N-terminal mutations anywhere else. As the ancestral ones, right?
Yeah, that's true. Especially for the emergence of the Kappa and Delta, we actually, I believe, we did not see any ancestral mutations in the N-terminal domain. N-terminal domain, unlike receptor binding domain, is not such a hotspot for studies. And it is very unfortunate. So because we all believe it's all about binding H2 receptor, but N-terminal domain certainly evolves very fast too. And it is known that it's actually virus evolves in N-terminal mutations accumulates during the infection too. So yeah, we really, the talk was heavily centered around the receptor binding domain indeed. Yeah, we need to take a closer look and what was it truly there were other mutations.
So any hypothesis on the impact of, or what you see in disease phenotype or something? How could this N-terminal mutations affect or, change the impact that are there already due to 452 mutations?
They can. The problem is, and as I said, that it's a mystery what N-terminal domain does. it's possibly that N-terminal domain actually very critical in the transmission itself, but after virus starting to infect a host, an individual, N-terminal domain might play lesser role than the receptor binding domain. However, it remains to be a target for antibodies. And it could be a target of antibodies that are neutralizing, opsonizing the virus. That's why it might be very important for the mutations to start accumulating the N terminal. Yeah, good. I see some questions. Do you want to read? I see some questions written here.
So first, let me go out to John. Sandeep asks, first of all, tells excellent presentation. Is there any direct correlation between virus spread and emergence of new variants?
I would say yes. That actually we see waves. If you consider new waves of the virus spread, the evidence for virus spread, it does seem that the waves are directed by new variants. Also, there is a very mysterious phenomenon which I didn't mention. It looks the virus is spreading in bursts, in like outbursts from single individuals who are called super spreaders. So it's much likely for virus to spread, not just by anyone who has the virus, infected with virus, but through specific individuals that are super spreading it. We don't understand, period, what it means. We know this is happening, and we know this is one of the major mode, if not the major mode of the virus spreads. But who are these individuals? Why they're so contagious? Is it possible that connected to specific type of individuals, or it's a transient status of anyone who is infected? Is it more virus spread? Is it more actively binding virus spread from these individuals? Are they spitting more when they're talking? Are they creating more aerosol or else? We don't understand. But it's clearly the case that virus primarily spreads through these super spreaders. Okay, I see another.
There is a question from Kamakshi. Kamakshi Sureka, I believe you remember her as well.
Of course I do.
So she first thanks you for one antibody cocktail will be effective against SARS-CoV-2.
Oh, absolutely. Absolutely. Antibodies, monoclonal antibodies are very effective. And frankly, against the variants, as we see, that might be actually the first thing what we will be able to come up with monoclonal antibodies against specific variants. And indeed, if we have a cocktail of the most critical neutralizing antibodies in hands against different type of structural variants, that actually could be at least a solution for us to avoid severe cases. Unfortunately, antibodies itself, if you inject them in the body, they are not living for long. They're stable for a week on average. Yeah, you have to reboost them again and again. Yeah, but yeah, absolutely, cocktail will work quite well.
Another question from Sandeep, it's kind of connecting. Is a recombination among viruses happening due to infection by different variants at the same time?
Most likely. I believe most likely. Yeah, it's a rare, very rare event. You know, not just they could co-infect, they should co-infect the same cell for this to happen. It is very rare event, but it does happen. Yeah.
Question from Rachana Banerjee. First of all, she thanks for the excellent talk. Question is these do these mutations have any correlation with any antibody binding sites of the protein that can help the virus in antibody escape?
Yeah, that's one of the major, truly hypotheses. Like the L452 mutations. One of our colleagues in Department of Biochemistry, David Wissler, published a paper. This one single mutation actually diminishing binding of 13 different monoclonal antibodies to the virus. And so the antibody escape seem to be one of the key parameters now why the variants are spreading. However, at the same time, you need to remember that the first variants that appeared, including the Kappa, including the Delta even, they appeared at the time when vast majority of population did not have any antibodies yet against the virus. They were not either vaccinated or they did not go through the infection itself. So it's hard to understand what was the selection. That's why people talking so much about increased affinity of binding, so the virus binds much better to cells. It's also possible that against these variants, the body is not producing very effectively neutralizing antibodies when the infection happens. So that's also, again, we don't know so many things about it. So everything's still on the table. Certainly the first variants, and possibly vast majority of them, appeared probably as a result of containment, our trying to communicate less, wearing masks, and so on. So it became more infective. So it takes lesser number of variants of the viruses to infect. That was the driving force originally. Right now, unfortunately, it could be the escape variants, immune escapes that could be driving the selection. Sujoy knows Shandim very well about all these evolutionary forces of the sweeps.
Yeah. So next question from Vidhi Agarwal. First of all, she thanks for the excellent talk. My question is that what are the challenges resulting in the delay in identifying the intermediary host animal for SARS-CoV-2, unlike SARS-CoV-1 and MERS, where both civets and camels were recognized respectively quickly as soon as the epidemic broke out?
I would say that actually we know quite well that SARS-CoV-2 came from bats. The SARS-CoV-1 and MERS, they were also coming from not identical viruses from the different animals, but from the very closely related ones. So all animals, all mammals, birds, they have their own coronaviruses, and diversity of coronaviruses within the same host species could be very, very high. So we only can guess that, yeah, that's probably the closest one to the bat viruses or camel viruses or whatever. But I don't think that SARS-CoV-1 or MERS actually was perfectly matched to any of the animal viruses. And then the fact that they were quickly identified what was the source probably did not play that significant role in containing the pandemic. What really was important for SARS-CoV-1, for SARS, the original SARS and MERS, that they did not, they always caused symptomatic infection. And it was much easier to recognize a sick person because of the symptomatic infection. And they were not as contagious as SARS-CoV-2. Problem of the SARS-CoV-2, primary reason for its pandemic, that it's spreading through asymptomatic or low symptomatic individuals. So we cannot isolate them very fast. It's important, probably important will be to find out where exactly it came from which rat, from what, but it now took its own sort of course of life, this virus.
Okay, questions coming in. I mean, it's already close to 11 p.m. If
That's okay.
Asking you, so yeah, thank you, sir, for such an illuminating presentation. I gathered so much new information. How can a recombinant virus, for example, SARS-CoV-2, invade new hosts, as we have seen recombinant from bat and pangolin?
No idea. Really no idea, because again, it takes so many things to come together to be able to invade a completely novel host. We're focusing a lot on the ability of the spike protein, for example, to bind to human cells. It's probably not, for sure, it's not the only answer. Virus itself, much more complex organism. It takes its replication. There is a replication rate inside the cells that matters, and so on. We really don't know much about it. You know, one of the good things that will come out from this pandemic, and already comes out, that we will start to understand much better not only physiology, not only epidemiology of pathogens, what does it take to emerge as a highly contagious pandemic, but also physiology of viruses specifically, not just coronaviruses, and how we can fight it. Yeah, it's a very good and a key question again, why this jump could happen. We are in contact with environment and animals all the time. Every day, hundreds of new coronaviruses contact us, but we only now we got a really bad one.
Okay. Another question from Shotati Shaha. So it's to you whether it's. Thank you, sir, for such an informative talk. Although today's talk was about SARS-CoV-2, but my question is for something else. That is, will you please share your insight about the probable evolution or evolutionary strategies that can be taken by cytophilic microorganisms if global warming takes place highly? What could be the probability of those evolution, such kind of evolution to cause disease in humans? Can we determine those probabilities by laboratory-based experiments?
Well, we can. It's a great question, actually. Yeah, there is some talks now how the climate change, if it's truly occurring, which I believe it does, yeah, so how would it affect the microorganisms that are way better, much more adaptable than high organisms, and if they will truly to become more temperature moderate tolerant, will we see new pathogens emerging from them. As of course, Sujoy Sandeep remember, well, my view is that vast majority of pathogens, human pathogens, are more accidental pathogens that really be able to cause infection rather than evolved to do so. So it is quite possible that new pathogens will emerge with planet becoming warmer, because they will start to adapt to a warmer environment and will start to like us.
Shundip's another question: can the infection by virus mess up the genome sequencing?
Not of this virus, I believe. No, you mean the genome? Can the co-infection by virus can mess up the genome sequencing? Maybe Shanip, you can mess up the genome thing. What do you mean? Can it affect the human genome? Or maybe if there is a multiple. Okay, that's what I see. Sorry, it's late here, so I'm not that fast. Yeah. Whether or not that we when we sequence and there is different viruses co-present, whether this could affect the quality of sequencing. It certainly can. However, usually one type of virus is heavily predominating, like in co-infections with bacteria. So you will see primarily sequence from one particular clonal group. And if you see them more or less equally present, you will start to see the calling of the position in the chromatography would be, or in the genome sequences, will be quite different. You will see multiple traces there. But yes, and it's actually a very legitimate question specifically for coronavirus, because we know, especially in the longer term infections, that virus starting to split into different subclonal groups, starting to co-evolve with during the infection. And indeed, it would be a challenge often to try to separate them from the specimen, clinical specimen. Ideally, you will have to go now to the cell culture and try to isolate a pure culture of viruses. But certainly, yes, that's correct, that there is this possibility, and that's a possibility that we actually might miss very important mutations by being masked by more dominant clones.
So, and Vidhi Agarwal, Shatabdi Shah, and Iftikhar Rahman, they all thank you for answering their questions and offering them insights late at night at your place, still being awake. So, one quick question, just out of curiosity, in your lab, I mean, as you, I mean, very interestingly found out a co-evolution with this receptor binding domain. I mean, this, not receptor binding, it's a different place, that 452 position mutation and the N-terminal domain mutations. So similar things, I mean, did you find anything, or are you analyzing other proteins as well, apart from spike, to understand any co-evolution going on under any kind of selection pressures?
Do you mean in the coronavirus, in the SARS-CoV-2?
Yeah, in SARS-CoV-2, apart from, I mean, just with and other proteins that there, I mean, nucleoproteins.
We do not do this, and this is unfortunate, because there is so much now focus on the spike protein because it's a vaccine target. And to a great extent, and we just disregarding at this point mutations in so many other proteins, including especially proteins that involved in the replication of the virus. Yeah, but it will come to that too.
So thank you once more to accept the invitation be here with us, and it's always a pleasure, at least for me and Sandeep, to see you even for a distance, to experience your smile and your talk, and whatever else. I don't want to be unconstitutional here when I'm live. Okay, so Sandeep, you want to end with anything, and so we can say bye to Evgeni and to all our viewers here.
Yeah, so again, thank you so much from all of us, from our chest family, GIS, IAS here, to, give us the time. And that was a really nice talk. We really enjoyed a lot. And thank you again.
Yeah, no, thank you very much again, Sujoy, Sandeep, for giving me the opportunity. And I just want to tell everyone, all the students who, or prospective students who are listening now present, you're really lucky to have people like Sujoy and Sandeep, the fantastic thinkers, and I'm sure they're fantastic teachers. Yeah, thank you again. And really good luck with your program.
Thank you.
Thank you.
Yeah.
All right. Bye.
Okay, so with this, we end today's meeting, and we'll meet you next on 21st of this month with our 5th lecture of